Hey everyone, my name is Jacob Allred, and today I'm going to be talking about the research paper I chose, which is Prey Shifts Drive Venom Evolution in Cone Snails. And I decided to choose this paper because I wanted to learn more about prey-specific selection pressures. With that said, within each venomous lineage of cone snails, there are interspecific variation in venom composition where several different factors have been proposed as drivers of this variation, whether it be phylogeny or diet. And so, because of their high species richness and the presence of major evolutionary prey shifts, these cone snails act as a great example for investigating drivers of interspecific venom variation. And within the study, the researchers analyzed the overall venom composition and individual toxin structures to try and demonstrate that the shifts from what they call vermivorous cone snails, which eat marine worms, to piscivorous cone snails, which hunt small fish, is complemented by distinct changes in venom composition that's independent of phylogeny. And with that said, the overall purpose of this study was to better understand the role of prey-specific selection pressures in shaping cone snail venom variation. And the authors hypothesized that prey preference shifts have shaped venom composition in cone snails. They also hypothesized that while all dietary groups of cone snails shared certain conotoxic gene groups, that differences may exist in the pharmacological class of what they call encoded conotoxins, depending on their prey preference. And so it's important to note that conotoxins are a group of peptides that interact with the nervous system to shut down certain ion channels within its prey. And I thought this image below did a great job of kind of showing the process of shaping venom prey specificity. You can see how it starts with a rich mixture of toxins and then decreases via purifying selection and then eventually leads to a highly potent prey specific toxin. So, in terms of the graphs that highlight the results of this research article, Figure 6A is showing when they injected venom of different cone snails, whether they were piscivores, molluscivores, which eat mollusk, or vermivores. What they did was they injected the venom of specific cone snails into a species of goldfish, actually. And as highlighted in the yellow and orange, which represent the piscivores, it caused the most decrease in movement over time in these fish compared to the molluscivore and vermivore species, which were not really affected as much in terms of the fish movement over time. And then figure 6B on the right represents, ancestral state reconstruction. And for the Geographus and Striatus, which are the piscivores, we see the highest oscillation movement change for the fish, indicating that, their venom composition is highly specific to the prey that they hunt. And then we can see with the molluscivores and vermivore species that they did not have as much of, they did not create an oscillation change to these fish as high as the piscivores. And then interestingly, we can see from when they conducted a PCA, which is a principal component analysis, which is basically a statistical method used to analyze physiochemical properties. And in this case, they were analyzing conotoxins. We see that for piscivory cone snails, when they were analyzed, that they were grouping in this area containing what they call mu conotoxins, which are a very specific conotoxin that shuts down muscle sodium ion channels within vertebrate species. So we know that this conotoxin is directly targeting small fish. And then in terms of the significance of the results that were found from the study, It's really important for understanding anti-venom therapy as well as, developing venom-based drug research. It's also really relevant for better understanding, the vast amount of conotoxins and the pathways that they take to disrupt ion channels, whether it be, sodium channels or others. And then also, this study is significant in terms of studying, ancestral cone snails in the process of um, their evolution, whether they're studying, venom ducts that are undifferentiated to uh, the undifferentiated of the worm hunter compared to proto compartmentalized to, uh, compartmentalized for the fish hunters. And so that sums up the majority of the study, and I hope you were able to learn something from this. Thank you.